Today I'll be discussing my senior research project on the effectiveness of various hand sanitizer delivery methods against bacterial elimination. Before we discuss the scope of my research, I would like to take a moment to tell you a little bit about myself. My name is Natalie Harris. I am currently a senior at George Washington High School, and in the fall I plan to attend the University of Virginia to major in biology on a pre-medicine course track. I chose this topic because the impact the pandemic has had on my high school career as well as my health, further causing me to take an interest in the prevention of bacteria transmission. Therefore, I will be testing different delivery methods of hand sanitizing products in hopes of finding the most efficient method to eliminate bacteria. Before I explain how I conducted my research, I would like to take a moment to elaborate on how others conducted their research. In order to understand how bacterial elimination occurs, it is important to take formulation of product into consideration. Hand sanitizer can be categorized into two basic groupings, alcohol-based and non-alcohol-based. Many studies have tested these bases against each other in hopes of finding the most efficient product for the general public. This has further resulted in the recommendation of alcohol bases. Further examining alcohol bases, a study in 2020 has found that ethyl alcohol proves to provide the most efficient bacteria elimination when compared to other commonly used alcohol bases such as isopropyl alcohol. In addition to this result, the study has also found that the concentration rate of this base has an effect on how well the product may work. This further found that the concentration rate must be 60 to 90% in order to properly break down pathogens and bacteria. Though this is not the only portion of the formulation that is important, studies have also found that fragrance may lead to skin irritation, respiratory complications, as well as less bacterial elimination. Relating to this irritation, it is important that hand sanitizers have a soothing ingredient such as glycerin to avoid damage to the skin. Glycerin is a humectant, which is a water-absorbing substance that allows moisture to be kept within the skin. While studying formulation, I found it necessary to find other concerns that may occur when applying hand sanitizing products. Research shows that alcohol bases tend to eliminate the most bacteria than any other commonly used alcohol base. However, without a soothing ingredient counterpart, they are likely to cause skin irritation, such as dryness of the skin, flaking, burning sensations, and rashes. As previously stated, to prevent this from happening, research shows and recommends that glycerin be added to product formulation. While there is a widespread of various hand sanitizer formulations that can affect how well a product may work, it is important to note the various delivery methods that can be used to apply these products. A research study has shown that foam hand sanitizers tend to eliminate more bacteria when compared to gel hand sanitizers. However, I have noticed that this is a common result when testing against gel alone, and there is little research comparing gel rub, wipes, and spray against each other. Considering how commonly these products are used, I find it's important that this be tested. To verify this trend, a review was conducted to compare alternative delivery methods of hand sanitizing brands. This review stated repetitively that these research studies were conducted with constant limitations of product. This further leads to the significance of my research and highlights the gap in the field in which I hope to assess. This gap being efficiency of Germix brand hand sanitizer delivery methods. The lack of research has led me to my research question: how do Germix brand hand sanitizer wipes, spray, and gel rub differ in the effectiveness of bacterial elimination on the hands? My null hypothesis was then formed and states if different delivery methods of hand sanitizing products are used, specifically gel rub, wipes, and spray, then there will be no difference in bacterial elimination. My alternative hypothesis states if different delivery methods of hand sanitizer are used, specifically gel rub, wipes, and spray, then there will be a difference in bacteria elimination. My independent variable for this study was the different delivery methods used to apply hand sanitizing product. My dependent variable was the amount of bacteria colonies counted after the application of hand sanitizer. My control was the amount of bacteria colonies counted before the application of hand sanitizer, and my constants include incubation temperature, participants, a clean working space, the length of treatment, and the brand of hand sanitizer. Before I began my procedure, I obtained consent from all participants, principals, superintendents, as well as parents. After this, I then gathered my materials, which included Germix wipes, spray, gel rub, sterile con swabs, and petri dishes containing nutrient agar. I then labeled my dishes both before and after, as well as giving them a corresponding identification number to each participant. The picture on the bottom right shows 1/3 of my sample the day it was collected, and the photo on the top right shows me swabbing one of my participants' hands before the application of hand sanitizer. I met with my participants in the Governor's School laboratory, being sure to keep my environment sterile. I then swabbed each participant's hand with a clean, sterile swab and streaked the swab against their corresponding before petri dish. This can be seen in the photo on the bottom right, where I'm swabbing one of my participants' hands. After all participants' hands were swabbed and they were streaked against all petri dishes, I then taped all dishes closed and incubated them for 24 hours at 37 degrees Celsius. I then swabbed each participant's hand after the application of hand sanitizer, which was applied for a time of 30 seconds, mimicking a study in 2014 that says 30 seconds of application proves to provide sufficient bacteria elimination. After this was done, these dishes were then taped closed and incubated for 24 hours at 37 degrees Celsius. After incubation, I then counted the number of bacteria colonies on each dish and took note of the amounts. This can be seen in the photo on the top right. The image on the bottom right shows a comparison of one of my participants' hand sanitizer spray dishes both before and after. The dish on the left is from before, and the dish on the right is from after. In order to visualize my data, I created a scatter plot in which I calculated the difference of all participants before and after bacteria colonies. This allowed me to account for the average mean of each treatment group. The mean of the gel was -16, the mean of the spray was -82.2, and the mean of the wipe was -29.3. The mean data shows that there is a slight difference across all 3 delivery methods. However, this is not as big of a significant difference to prove that there's a difference between each delivery method. Viewing this graph, it is also clear to see outliers within my participants, which cause the difference in mean, standard error, and standard deviation. To see an example of one of my major outliers, I've highlighted it in the green box. This is my largest outlier of -467. This outlier causes the average mean to be significantly lower than the other treatment groups. This further means that the lower the average, the more elimination that occurred. This also suggests that my spray data was skewed due to outliers within my participants. The statistical test I used to interpret my collected data was a one-way ANOVA test. This allowed me to compare p-values of all my treatment groups. These p-values consisted of 0.59, 0.98, and 0.71. These values show that my data was insignificant because they are greater than the significance level of 0.05 that they were testing off. This further supports my null hypothesis. The reason that these hand sanitizers perform similarly is because they have the same formulation. A similar study has the same outcome when they tested against a different German brand. The significance of my research leads to the conclusion that hand sanitizer delivery methods such as gel rub, wipes, and spray are similarly effective, and no one method should be used over the other when seeking more bacteria elimination. The limitations of my study include my sample size and my range of product. Although my sample size was 36, I still consider this to be a small sample and may limit how accurately my results represent the population. Also, the fact that each treatment group was applied individually leaves room for human error and the lack of efficiency of treatment. Lastly, only using one brand of hand sanitizer limits my ability to compare this brand to other brands that were not addressed within my study. In addition to only using this brand, I only chose to use 3 delivery methods of hand sanitizing product. I have found that the 3 methods I tested, gel rub, wipes, and spray, have similar effectiveness of the elimination of bacteria. However, future research should test if these methods, compared to other methods that were not addressed within my study, such as foam and liquid hand sanitizers, have any difference of bacteria elimination. In addition to this, there could also be a possibility that a different brand of hand sanitizer has a different outcome than the Germix brand within my study. Finally, future research should also test with a larger sample size in order to have a better representation of the population corresponding with the outcome. Even though my research hypothesis was incorrect, going through the experimental process and learning how to properly perform research and lab experiments allowed me to pique my interest in biology, which allowed me to choose my major in college. At this time, I would now like to thank my participants for taking part in my study and making it possible. I would also like to thank Ms. Long for all the assistance throughout the entire writing and experimental process. And lastly, I would like to thank Ms. Dillon for all of her help with the laboratory technique and the creation of my petri dishes. Thank you, and are there any questions? Ms. Martin?
I apologize if you said this and I missed it, but did all 36 of your participants undergo each type of hand sanitizer?
Ms. Martin asked if all 36 of my participants went under the same types of hand sanitizer. I only had 6 participants within my study. However, each participant did a before dish and an after dish, which all added up to 36 in the end, but yes, all of them did undergo the same treatment.
Okay, thank you.
Mr. Taylor?
How much instruction was given to each participant for the application of the gel, the spray, or the wipe, or did they just do it to their own fashion?
Mr. Taylor asked how much instruction was given before the application of hand sanitizer for each treatment. I provided my participants with instructions the first day of treatment, and I asked them to repeat it on every testing day.
I have one more. Was there any differences between the concentrations of active ingredients amongst the 3 types that you tested?
Mr. Taylor asked if there was any difference of concentration rates between my 3 treatment methods. The only difference that I had in my research was that my spray hand sanitizer did have a scent because there was no unscented version.